So welcome to the last section, the last lecture, and the last lab of our course. And then hopefully you will know most of what you need to know to analyze your own data sets to a standard where you can publish them in a modern day publication. Okay, so this section is based on our course, edited then by Gary, and then Gary Bader from Toronto, and then edited again by me for this iteration of our course. Very much a collaborative lecture here. So Gary wrote some learning objectives. I'm just going to skip over those though, because you'll see what you're going to learn. That's what the whole lecture is for. So this whole lecture is based on integrating your data. So this is the case where for our modern day single-cell RNA-seq data sets, one sample isn't enough. We've got to have multiple samples across multiple conditions. So how do we work with that? So the main way we're going to do this is by taking the cells from multiple samples and combining them together to do a joint analysis. So we're going to merge them together and then cluster them and annotate them all together. And then we can compare between our different conditions in each of our cell types. This sounds great, and it would be awesome if it was really that easy, but we have some problems in that we can have pretty severe batch effects between different data sets. So here's an example of a data set of PBMCs sequenced with 3 different protocols from 10x. So we have 3' version 1, 3' version 2, and 5' single-cell RNA-seq. Oh, let's move you guys. Where's my control? There you are. Let's pull you down over here. Oh, you're going to pop up there. I'll put you down there for now. Oh, there's a way to hide it, isn't it? Yeah, can I do that? Yep. There we go. There we go. That's better. Now you can see the slides. Yeah, so you can see there's pretty severe batch effects. Our purple, orange, and yellow cells from our different technologies aren't mixing together at all when we join them together. Instead, we get 3 sets of clusters where we get each of our cell types in 3 separate clusters, one for each technology. So you can see we've got a cluster of T cells for purple, a cluster of T cells for green, and a cluster of T cells for orange. This is not ideal. We would much rather have all of the T cells grouped together because they're all the same cell type. They should group together. So we can use a batch effect correction method or an data integration method. People use those terms interchangeably, even though, like, technically, as we'll get into on the next slide, I would prefer there to be a difference, but they're often used interchangeably. And we're going to use, in this case, we used Harmony. And now you can see our orange, green, and purple cells are all mixed together, and we get one cluster of T cells, one of monocytes, one of B cells that are the same across all of our different data sets. Okay. So we fixed our batch effects. Perfect. So what happened here? What are we actually doing? So there's a few different ways of joining your data together. The simplest is just merging your data. This is just taking your data sets and putting them together. So instead of having one matrix for each sample, you now have one matrix with all of your samples, and you haven't done any sort of batch effect correction or data integration. You've just merged them together. You might have done some normalization before you merge them together, but that's about it. You can do batch effect correction, which should really refer to when you're actually correcting the gene expression values to get your data sets to merge together, and you're actually removing the batch effect from your gene expression values. And there's your data integration, where you're merging together your samples, but you're not changing the gene expression values. You're just changing the lower dimensional space where you're projecting your data into. So you're changing your UMAP, you're changing your PCA, but you're not changing your gene expression values. That's how I would define them. In practice, people use batch effect correction and integration kind of interchangeably in the literature. So when do we need to do batch effect correction? So first, what is a batch effect? So batch effect is defined as random, unexplained differences between batches of samples. So these might have been samples that were run on different days, on different machines, or by different students, and they look different to each other, and we don't know why. We assume when we call something a batch effect that this is a technical difference. So it might have been, oh, the temperature was different on one day versus another day, and that affected the stress level of our cells. Or maybe we used a different set of reagents. One was older than the other, so we got different quality of cell lysis and reverse transcription, right? So it's a technical difference. And the problem is when we have really strong batch effects, it interferes with our clustering and finding trajectories in our data, right? We'll get separate trajectories or separate clusters that represent the same cell type in different batches. It also makes our visualization really hard to understand because we've got duplicates of everything all over the place. It can also reduce our power when we're using differential expression analysis. So if we're comparing our disease versus control, if we've got a lot of batch effects between our samples in each group, we're going to have a lot of noise within each group, and it'll be harder to detect differences between the groups. There's a bunch of ways you can measure whether you have batch effects in your data and whether batch effect correction is needed. So there's a whole bunch of scores. You can look these up on your own if you want to know exactly what they are, but they're all essentially trying to measure how well mixed are the labels of your different batches versus how well mixed are the labels for your different cells. So if you have no mixing between your batches, that's bad, and you want to do batch effect correction. If you have lots of mixing between the labels of your different batches, you don't need to do batch effect correction. They're already fine. You can just merge them. And while you can calculate all of these scores and try to come up with some sort of threshold, in practice, it's usually pretty obvious if you need to do batch effect correction. You can just throw up your UMAP, color it by your different batches, and say, oh, no, I've got duplicate clusters for all of my different batches. So I need to do batch effect correction. So in practice, usually what we're doing is just look at the UMAP. Are my samples separated? If yes, I need to do some sort of batch effect correction or integration. We might also look at whether this is a systematic shift in all of the clusters, or is it specific to certain clusters where we have what looks like a batch effect. That could also influence our decision to do data integration or not. And you might be asking, why do we even need to check? Why don't we just always do batch effect correction, right? If we have different batches, just always do batch effect correction. The reason we don't is because whenever you're changing your data, you're losing something. So if we do batch effect correction when we don't need it, we could actually lose biological signal that's meaningful in our data. So we don't want to do it unless we absolutely have to. And our batch effect correction methods can make mistakes. So here's some data we actually published, and the reviewers didn't notice that there's this glaring error because of our batch effect correction, where this is cells from the liver, and in pink there at the end of the blue cluster, so the blue cluster are all hepatocytes, that pink cluster is B cells. B cells look nothing like hepatocytes. And yet here on our UMAP, they're stuck together, and that's because we used a very strong data integration method, and B cells were not present in some of our samples. So our integration method said, hey, what's most similar to a B cell in the sample? It's a hepatocyte. Okay, stick together, you're now the same. Okay. And it's clearly wrong. Like obviously, no, B cells shouldn't be clustering with hepatocytes. So you do need to be careful. They can do incorrect things. So here's another example. So this is also liver. And in this case, we see a whole bunch of clusters that are different cell types. If we color this by what sample they originate from, you can see that a lot of the clusters have good representation of our different livers. So you can see like cluster 8 over here, we've got cells from at least 3 of the different livers. Same with cluster 2 and cluster 9. But we also have some clusters like cluster 5, cluster 1, 3, and 6 that are essentially completely unique to one of the samples. So do we need to do batch effect correction here? Or is this a real biological effect? Because these are human samples and we're looking at liver, well, people don't all have the same kind of liver, right? Someone who drinks a lot will have different genes regulated in their liver than someone who doesn't. Someone who's a vegetarian versus someone who eats a lot of meat. So it could be that these differences between different livers is expected, and it could be real biology. Or it could be a batch effect. So what is it? What are we going to do? Right, so the differences could be real biology. Could be different genetics between different people, different diet, different age, different sex. Or it could be a technical effect. So it could also be a technical difference. These are from human samples. They were dissociated on different days by different students across about 5 years. So also the reagents were different. So it could also be technical differences. And just by looking at our UMAP, we don't know which is which. So what did we do here? So the reviewers asked us, of course, how did you control for batch effect in your experiment? So what we did was we attempted a whole bunch of different batch effect correction methods, including explicitly regressing out technical factors like what student did the experiment, the sex of the individual patients, the library size. And no matter what we did, even using some integration methods like canonical correlation analysis and mutual nearest neighbors, we're going to talk more about those later in the lecture, we still didn't get those hepatocyte clusters to merge together. So we concluded that these represent true biological signals. These livers are different between these different people for real biological reasons. We're just not sure exactly what those biological reasons are. So we did not end up doing batch effect correction for this data set in this paper. Here's another example. Is this a biological effect or a batch effect? Here we have 29 glioblastomas, and we're only looking at the tumor cells. And on the left, they're colored by sample, and on the right, they're colored by the clusters we get when we do our standard single-cell RNA-seq analysis pipeline. And you can see again that the tumor cells aren't merging between different individual samples. Right? We have approximately 1 cluster per tumor here. Is this real biology, though? Tumors have lots of different genetic variation, right? Each tumor requires a different set of mutations. This could be real differences because of the different genetics or different driver mutations in each of these different tumors. But they could also be different technical differences. These tumors came from a biobank. They had been stored there for different amounts of time and preserved in slightly different ways. So it could also be a batch effect. So what did they conclude? So the reviewers asked them, how did you control for batch effect in your experiment? Is this technical noise or is this real biology? So they also applied 3 different batch effect correction methods. None of them were successful in uniting the clusters. So they concluded this is real interpatient heterogeneity, and it's biological in nature. So these are different driver mutations driving these tumors, so they really should be different. However, that's not always the right answer, right? So both of these papers came from the same lab. They were both looking at human livers. One of them did not use integration, and one of them did use integration. So there's no universal answer. So the reason we used integration on the right is because in that paper, we did 2 different technologies on these cells. So we did both single-cell RNA-seq and single-nucleus RNA-seq. So we knew the differences between single-cell and single-nucleus were technical in origin because they were on the exact same patients. So we did integration in order to merge together our single-cell and single-nuc data. Whereas on the left, We mostly saw differences only in the hepatocyte cluster, and we believe that's real biological differences. So how do we determine we've done a good merge or batch effect correction? So how can we verify our batch effects are removed? So one is to just look at the UMAP. This is the most popular way, but it can also be the hardest way to really judge, because often your cells will be stacked on top of each other. So it can be hard to see exactly how much contribution you get from each of your different batches to each of your clusters. You can also check whether your clusters make sense and whether they represent a single cell type. And each cell type is represented by a single cluster. Right? So if you have well-integrated data, you should get one cluster per cell type and one cell type per cluster. You can check whether the biological signals you expect to see in your data still exist. So if you've done too much batch effect correction, like you saw with the B cells and the hepatocytes, you can see whether your biological signals are stopping to make sense. So do you have cell types clustering together that really shouldn't? That it makes no sense for them to be clustering together. Or do you have something like cell cycle effects that are now all mixed together with non-cycling cells? And then, and then making sure the interesting biological signals are not ruined. So your different cell types are still distinct. Your cell subtypes you're interested in are still distinct, and they haven't gotten all mixed together from your data integration. And then the best answer, of course, is to check all of these things. So never rely on just one measure. Check everything, make sure your data makes sense, and it's not obvious that something has gone wrong. So in this case of our 5 healthy human livers, we saw visually that we have significant mixing of different samples for most of our clusters, which means it's unlikely to be batch effects. If we then visualize this as a bar graph where we see the contribution of each sample to each cell type we annotated, we can again see that most of the clusters are found and have cells from all of the samples. So again, suggesting our batch effects, it's unlikely to be batch effects. If we look at our cell type annotations, we can see that most of the cell types have only one cluster associated with them. So all of the immune cell types, the gamma delta T cells, the NK cells, the CD4 T cells, they were all represented by a single cluster. So again, batch effects are unlikely. Whereas if we look at our PBMC data set, here visually, the batches are completely distinct from each other for all of the cell types, meaning batch effects are likely in this case. If we look at the contribution of each sample to each cluster, we can see that almost all clusters are unique to a single data set or a single sample, again meaning batch effect is more likely to be the explanation. If we look at the cell type annotation across our clusters, we again find that in many cases, for most of the cell types, they're split across multiple clusters, again indicating this is more likely to be batch effects. So in this case, we would want to do batch effect correction on this data set. And once we have done it with Harmony, we can check again to see if this has fixed everything. So visually, after doing Harmony, it looks pretty good. The samples look like they're overlapping each other for most of the cell types. If you look at the frequencies of the different samples across the clusters, now every cluster has contribution from every sample. Looks like we fixed the batch effect problem. And if we look at the cell types across the clusters, now most of the cell types are represented by only one cluster. So again, it looks good. Looks like we fixed our batch effect problem. right? So that's how we can tell our batch effect corrections methods were effective or not. So we always have to be careful. It's already mentioned that in some cases we can overcorrect and mix together cells that shouldn't be mixed together. So overcorrection in this case is defined as the removal of interesting biological variation that is of interest. So maybe we've removed the cell cycle signal when we were looking at cancer, and our cycling cells are now mixing together with our non-cycling cells. It's probably not what we wanted. We might have cells of different cell types or states mixing together and no longer forming distinct clusters, which is, again, not what we want. We may also see samples of different types being merged. So if we have tumor tissue and tumor-adjacent tissue, if we see that tumor-adjacent tissue merging together with the tumor cells, that's probably also a bad sign, right? Those probably should be kept separate. So you need to be very careful when you're doing data integration to make sure you're only getting rid of the least amount of information you need to in order to get your data to integrate. And there's no trick to this. There's no algorithm you can use that will tell you whether you've overcorrected or not. You just have to know your data and know what it should look like. In terms of undercorrection, this is when you fail to remove all of the batch effect. And that's when we do have some scores that you can use to calculate whether there's still some batch effect present or not. Different methods for doing data integration are more or less likely to over or undercorrect. This tendency can be described as harshness or strength of the integration method. So you might see harmony described as a strong integration method, which means it has a tendency to overcorrect rather than undercorrect. Whereas you'll see something like RPCA being described as less harsh, which means it is less likely to overcorrect and more likely to undercorrect. So how does batch effect correction really work? So the first step is always to identify some sort of common biology between the batches. Depending on the method, it might have a statistical way of doing this, or it might ask you for it. So it might ask you, give me the labels for the different cell types for each cell. So usually this is done by looking for similar clusters in the data. So here we might find that all of these groups of T cells and NK cells, they're all similar to each other. You can also look for similar cells. So you could look for individual cells and what their most similar other cell is in the other batches. So here we might see these B cells, individual B cells, match each other across the different data sets. You can also look for similar factors, which is essentially looking at your principal components and asking how similar are my principal components to each other between my different data sets, and finding those principal components that are similar to each other. The next step is to calculate the difference between the batches for that common biology. So if you're looking at pairs of cells, what's the difference between the pairs of cells from different batches? If you're looking at different factors, what's the difference between those factors in the different batches? Once you've calculated what that difference is, you just remove that difference. And that's all there is to it. Then batch effect correction and data integration is used interchangeably, but usually batch effect correction is thought to be changing the data to remove technical effects, whereas people will use data integration when they're talking about just combining multiple data sets together by aligning their biological signals. So the difference here is we're not explicitly assuming in data integration that the effects we're removing are technical. they could be biological. Right? So if we have different samples from different people or from different tumors, we could integrate those together if we really wanted to. But then we might be removing differences that are real between our different individuals, right? Real biological variability between our replicates. In single-cell RNA-seq, batch effect correction and data integration are treated synonymously, which is very annoying. Or they're used to refer to how the correction is applied. So whether it's applied to the lower dimensional space or the expression values. Or they can be used to refer to the different scale at which the data is being combined. For instance, combining multiple data sets from different publications and research groups can be called data integration, whereas within a single experiment, you might call it batch effect correction. It's very irritating that scientists don't use their terms rigorously, but that's how it is. People use terms how they like. So whenever you're reading a paper and they're saying they're doing one versus the other, always check exactly what it is they're doing, because they could be doing various different things and calling it the same thing. In many cases, most of the batch effect correction methods treat batch effects as a factor in your data. So a factor, if you remember like high school math, a factor is one component of something that you can multiply together to get a number. So a factor of the number 6 might be the number 2. So you can do 2 times 3 gives you 6. This is the same sort of concept we're using for factors here, but here we're talking about them in the context of a matrix of data. So we're also talking about these as biological factors, right? So these confounding factors are generally nuisance sets of variability that could be biological or technical in origin. It can represent a physical process, such as the cell cycle, that affects many genes. So we already talked about confounding factors, right? Things like the cell cycle, genetic environment, genetic background, environment, lifestyle, age, etc. that we talked about previously. Yeah, so in math, a factor is a factor of a number that you can multiply together to get the original. That's the same thing we're doing when we're talking about factors in single-cell RNA-seq, only now it's a factor of a matrix, meaning that you can multiply the matrices together to get back the original matrix. There's lots of different ways to calculate factors of a matrix. We're not going to go into all of them. If you're really interested, you can look those up, but I suggest you have at least one linear algebra course under your belt before you try and do that. So the main example we've been using so far is PCA. So PCA does matrix factorization, and your different principal components are your different factors. If you take your sets of matrices you get out from PCA and multiply them together, you'll get back the original matrix. Right, so we already talked about PCA. So here's another example of some data where we would pull out PC1 along this axis and then PC2 along that axis. So most data integration methods that are used today align related factors within your data. So they don't actually correct the gene expression values, and we'll talk about why that is in a little bit. So we're assuming that a significant portion of the biological variability in each of the data sets overlaps with the others, so we can align, so there is something to align between the data sets. So they might, the main situation here would be that the data sets must have overlapping cell types. So it makes no sense to try and integrate 2 data sets that only overlap by 1 cell type. It won't work, and it is not worth your time to try to do it. You want to have multiple cell types that overlap. There's lots of different ways of calculating factors. Most of the methods we're going to talk about are linear methods, but there's also nonlinear ones. You can use deep learning to find these factors. There's lots of ways to do it. They can be identified separately for each data set and then compared to each other, or you can use algorithms that try to find them jointly across the data sets. In many cases, though, the data integration tool won't tell you what these factors are. It will just spit out at the end the corrected data set. And you'll just have to trust it that it did something sensible along the way. All right, so which data integration methods should you use? If you go looking in the literature, there's over 30 of them out there. There's some benchmarking studies you could look at if you want to look at which ones work well. But there's no theory that tells you which one to use. And in fact, there's a theory that tells us we can't ever have an answer for what one is the right one to use. Because fundamentally, when you're trying to optimize something like the alignment of two data sets, the best way to do that will differ depending on what specific data you're talking about. So fundamentally, if you try to look for what's the best across all possible data sets, there is no correct answer. So in most cases, people end up just trying a few until they find one that works for their data set and stick with that. There are data integration benchmarks that you can look at if you want some guidance on where to start or what method to use. We can also learn on experience, so you can come talk to someone like me or Gary who does this all the time and ask, which one do you think we should use? Because they've probably used all of them multiple times and know which ones are easy to run and tend to work well. So in answer to that, since I'm here already, the one that tends to work well and fast and is easy to use is Harmony. So Harmony essentially is cluster-based, so it takes the cells in each of your data sets. So if you look over here, we have each of our data sets marked in different colors here, and the different shapes are different cell types. So Harmony tries to form clusters between the different data sets. It then assigns each cell to what cluster it was assigned to. It finds the middle of those clusters in each of the data sets, and then it just moves those clusters together in the lower dimensional space. And it keeps doing that over and over again until it stops moving things around. And you get nice homogeneous clusters that have cells from all of your data sets mixed together. It's considered a harsh or strong method, so it will almost always successfully integrate your data. However, if you have cell types that aren't found across all of your data sets, it will do the next best thing, right? It will still try and find a cluster that spans all over your batches, so it might make mistakes and force cells that aren't the same cell type to cluster together if a cell type is completely missing from one of your batches, as I showed previously with our data set where that happened. Another option is mutual nearest neighbors, which is kind of on the opposite end. So this one is not very harsh, but it's more careful to not mix together things that shouldn't be mixed together. So it's based on the concept of mutual nearest neighbors. So here we have our one batch up here, our second batch down here, and the colors indicate the different cell types. So if you find mutual nearest neighbors, which are take each cell in this batch, find what's the most similar cell in the other batch. And then for this batch, find that for each cell in this batch, find the most similar cell in that batch. And if a cell in batch 1 says, hey, you in batch 2, you're my neighbor, and that cell says, yeah, you're my neighbor too, your mutual nearest neighbor, and that will line up which cell types are actually matching. So it means these red cells will match with these red cells, and these red cells will match with those red cells. So they'll agree and say, yes, we are mutual nearest neighbors, we are the same cell type. But if you look at this yellow group, that yellow group doesn't have a good match in the other batch. So that yellow group will say, hey, blue group, I think I'm most similar to you. And the blue group in the other batch will say, no, 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 we're most similar to the blue group. So they won't get a match. So it will be able to tell apart cell types that are unique to one batch versus the cell types that are shared between the batches. And then when it goes to calculate its correction vector, so how to do the correction, it only uses those cells where there are matches. So it won't force together this yellow group to mix together with another group in the other batch. It will recognize that, no, that should stay separate. So when you integrate them together, you get the reds and blues mixing together, but the pink and yellow, they stay nice and separate. So that method was developed by this group down at the bottom, Harity and Nature Biotechnology. It worked so well that the people at Surat were like, yeah, that works great. We're going to borrow that for our method too. So now the modern Surat canonical correlation analysis also uses mutual nearest neighbors, but they call them anchors instead. But whenever you see anchor in Surat, it means mutual nearest neighbor. So canonical correlation analysis is a variant on PCA where it tries to find a shared space between two data sets. This makes it a little bit harsher than your standard mutual nearest neighbor method. So it can misalign data sets when there's very large differences in the cell type frequencies. So Surat now recommends using reciprocal PCA instead, which again uses mutual nearest neighbors. Only now to calculate those mutual nearest neighbors, instead of doing the CCA to find a joint space, it takes the PCA of one data set, calculates the PCA of the other data set. It takes data set 1 and projects it onto the PCA space of data set 2. It takes data set 2 and projects it onto the PCA space of data set 1. And there, within each of those joint spaces now, calculates the mutual nearest neighbors. After that, it does exactly the same thing as CCA does, where it finds this joint embedding space and then aligns the anchors to each other to calculate how to do the adjustment across the data set. The last one that takes a different approach is Liger. So Liger doesn't bother so much with aligning all of this biological data together. It throws all the data together, does PCA, or in this case, non-negative matrix factorization, which is a slight variation on PCA. That's again, decomposing the matrix into two components or multiple components. And then it just tests each of those components to see, is this component unique to one data set? Or is it similar across the two data sets? And then if you want the joint embedding space, it just takes all of those factors that are shared and gives you those and leaves behind the ones that were unique to one data set. And then if you're doing an analysis where you want to look at the variation that's unique to each data set, it goes and gets you those that are unique and gives you those instead. So it doesn't really do a correction. It just subsets the lower dimensional space into bits that are shared and bits that aren't. The last major approach to integration is reference-based integration, which arguably isn't really a different method. It's not a different algorithm for doing it. It's just instead of taking two data sets and considering them equal to each other and trying to mix them together, you take one big good data set as your reference, and then you try to map your new data set onto that. So here you're mostly keeping that reference data set the same, and you're mostly just changing your new query data set.
It's using another reference control or just using one set of outsamples as a control, then just change the other one.
Yeah, so the question is whether we're using an external reference or using one of our samples. So it can be either. So most often this is using an external reference, but for certain people and certain projects, they also actually use an internal reference. So maybe they did a bunch of samples at one point in time that were really good, and then they build on top of that and add more samples to that project. Yeah, so there's a couple methods for this. So Surat, people who make Surat made a tool called Azimuth that does this specifically for PBMCs. So if you have a PBMC data set, you can use their Azimuth tool to map onto their reference data set. And there's another one as well, Symphony, that can do this. But again, this relies on having that reference data set that you're mapping onto that's already annotated. We can also evaluate the performance of these methods. Yeah, so someone in Ottawa has used Azimuth. They say it's not great for monocytes. So yeah, it can work, but it doesn't always work. So you also have to be careful. Yeah, so when we're evaluating the performance of our batch effect correction methods, generally we're looking at, do they remove the batch effects and do they preserve the cell type differences? So the two goals we want to have at the same time. There's a bunch of benchmarks that have been published. So here's one of them. They say Harmony, Liger, and Surat version 3 are the best. Harmony is recommended as the first try because it's very fast. Here's another one that looked at different tools in Python. So this is mostly useful if you want to do an atlas level project. So if you're looking at dozens and dozens of samples, then you'll want to do one of these Python-based tools. Otherwise, you will require all of the RAM in Canada in order to load your data sets. R is very memory inefficient. Python is much better. So if you're working with more than 200,000 cells, I highly recommend moving to Python rather than working with that in R. Yeah, that's a whole other workshop for that. But in terms of concepts, all the same. So the conceptual steps are the same as we've been doing in R, just the commands you'll be using to run those steps are different. Here's another one that did both Python and R. They recommend the mutual nearest neighbors methods, the SCVI method, so that's a Python method, and Liger. So they said, actually, no, those performed poorly. The ones that performed well were Harmony. Yeah, so they just recommend Harmony. They found Harmony worked the best out of all of them. That they found MNN, SCVI, and Liger under-corrected, whereas Combat, PBKNN, and CCA introduced artifacts. So they overcorrected, whereas Harmony they found was the best. There's another table where they compared all these methods on a whole bunch of different metrics. I probably can't really read them that well. But in general, they find that SCANVI, which is a Python method, works well. And out of the R-based methods, they find that Harmony and yeah, they find that Harmony works the best out of the R-based methods. But mostly they find that the Python methods are better. I'm sure that has nothing to do with the fact that this whole lab that published this method is like, yes, everyone needs to switch to Python right now. And they run a whole set of workshops for, yes, here's how you do single-cell RNA-seq analysis in Python. So if you want to learn how to do it, that's the group you want to look up. They have lots of tutorials for working in Python. They are Python advocates. Yeah, so then we have a note on very large data integration tasks. So you need a lot of RAM to run data integration. You may need to use Python if you're working with large data sets, as it's much more efficient than R. And there's lots of research ongoing to improve integration, especially with deep learning, because AI will save the world and is the best thing for everything ever. Of course. I'm not biased. Yeah. So then the last thing I want to talk about here is this controversy with respect to calculating differential expression after integration. So you might think, hey, if there are all these technical effects that are causing noise in our data, we should get rid of those to improve our differential expression test, right? Seems very natural and logical. The problem with that is that almost all of these integration methods and batch effect correction methods don't distinguish technical versus biological differences between samples. This means that in single-cell RNA-seq, our batch effect, and because in single-cell RNA-seq, our batches typically also represent a single biological replicate, you can't tell apart what's a batch effect and what's a difference in the biology, right? It could be a difference in the biological replicate versus a different biological replicate, or it could be a technical effect. If you correct for the differences between biological replicates that are due to the noise between replicates, you're artificially inflating the significance of your differentially expressed genes. So you don't want to do that, because that will create false positive results. As a result, for the most part, integration methods in single-cell RNA-seq do not correct the gene expression values deliberately, because they don't want people to start doing differential expression on the corrected counts and getting false positives. Another reason this is a problem is that many differential expression methods assume the expression values follow a specific distribution. So they usually assume it follows a negative binomial distribution. If you do batch effect correction to your gene expression values, it will no longer follow a negative binomial distribution, and your differential expression method is now invalid and will not give you correct p-values or estimate your parameters properly. So again, you don't want to be doing DE on corrected values. So how can we account for batch effects in differential expression? Most of the time, we just don't. We leave them in, we assume they're mostly biological in origin, and treat them as real noise between replicates, because that's safer. If you assume that, then the worst case scenario is you miss something. You're not going to get false results. If there are known technical factors, such as different technologies, those can be included in models if you have sufficient replicates with each of those technical factors. So if you did like half of your data set with 3' single-cell RNA-seq, half of it with 5' single-cell RNA-seq, and you have replicates of each of your biological conditions with both techniques, you can add that technical effect into the model for your differential expression test, and that will correct for it. In theory, as far as I know, no one has actually tested whether that works or not, but it should do. So if you want to do differential expression across data sets using different technologies, you want to have at least 3 samples in each of your biological conditions in each of your technologies to do that reliably. Okay, to summarize, we talked about verifying that batch effect correction or data integration is needed before applying it. Don't apply it if you don't need it. Often, so I'll say, if you're working with mouse data, usually you will not need to do data integration. If you're working with human data, usually you will need to do data integration. Because, again, these are actually not technical noise effects. These are real biological noise. So this is genetic differences between people, lifestyle differences between different people. It's not that the reagents in my 10x experiment was slightly different between this sample and that sample. Because in mice, you don't see batch effects. Yeah, so I was asking, yeah, don't do batch effect correction before DE. Yeah, so you can do it before DE, but when you do your DE, you need to go back to the raw counts. So you need to save your raw counts somewhere so that you can always go back to them when you want to do differential expression. Batch effect can be corrected and removed, but you have to check the results to make sure it didn't remove other stuff that you wanted to keep. Overcorrection and undercorrection are possible and common. So we've done many projects, and we've seen both of these in at least one of these in at least 50% of them. So it's very common to have overcorrection or undercorrection, depending on your experiment and what method you used. And when you're selecting an integration method, you may have to try multiple ones. But it's suggested by multiple people and multiple papers that harmony is a good place to start, because it will usually work. And if it works for you without erasing your biology, you're basically good to go. And I have our fun fact that even though lots of people claim single-cell RNA-seq was invented in 2007, somewhere in the US, it was in fact invented in Toronto in 1990. They did the first RNA-seq of a single cell by hand, picking one cell out and putting it into a well and doing the experiment. So I'll take any other questions about data integration before we move on to the lab. I don't see any questions in Edmonton, so Edmonton can open up their RStudio terminal. Oh, we have a completely out of topic question from Ottawa, which is integrating different omics data sets. So this is another case where it's very annoying that people use the word integrating for lots of different things. So integrating different omics data sets, it depends on which omics data sets you're talking about. So for bulk RNA-seq and single-cell RNA-seq, usually what you're doing is deconvolving the bulk RNA-seq using your single-cell RNA-seq. And that will essentially let you estimate the contribution of different cell types to your bulk samples. Otherwise, you can compare the log fold changes of different genes and then see what cell types are most express those genes and sort of guesstimate which cell types are most likely to be changing. Okay, I don't see any other questions.